DNA inside the nucleus contains the instructions to make proteins, but the protein-making machinery is outside of the nucleus. The instructions are transcribed into a molecule that can leave the nucleus, messenger RNA, mRNA. mRNA exits the nucleus via the nuclear pores. It then heads towards the rest of the organelles that are in charge of manufacturing, processing, and packaging proteins. The nuclear pores are seen here in their open and closed configurations. The endomembrane system is involved in the production and transportation of a cell's proteins and other products or wastes. The organelles of the endomembrane system include the nuclear membrane, the endoplasmic reticulum, the Golgi apparatus, transport vesicles, the cell membrane, and lysosomes. These membrane structures form compartments that are all connected with one another. Endo means within, membrane means thin barrier. Here is a diagram of the endomembrane system. When RNA leaves the nucleus, it enters the endoplasmic reticulum, or ER. ER is a series of membrane-bound sacs and tubules and is continuous with the outer membrane of the nuclear envelope. There are two types of endoplasmic reticulum, rough and smooth. Rough ER has ribosomes attached to its membrane, thus a rough appearance. These ribosomes synthesize proteins that will be used in the plasma membrane, secreted outside the cell or shipped to another organelle called a lysosome. As proteins are made by the ribosomes, they enter the lumen, or opening, of the ER, where they are folded and processed. This picture shows proteins anchored in the ER membrane, but other proteins may float free inside the ER lumen. The ribosome is made of ribosomal RNA, or rRNA, and proteins. This is where translation occurs. Ribosomes consist of two subunits, a small and a large one. Each subunit consists of proteins and rRNA. The two subunits assemble around an mRNA molecule when proteins need to be made.